So I was thinking on how when I was thinking on how to do this talk, I had several ideas. So I hope the one I chose works. Again, thank you very much, everyone, for being here. Um, this is the first event I attended for the Systematics Association. It was 10 years ago, almost 10 years ago. It was a joint event with the Linnean Society. I think it was on systematics and illustration, and it was a homage to Rosemary Wise here, who is the botanical artist at Oxford, and that's me in the early days of 2015. This was my first contact with the association, and this was my last in-person event uh, in the association with the association, at least for now, until the next one. This is the council meeting we had less than a month ago at the same place, Linnean Society. Um, we had a council meeting that's on the top left, and I took another picture of myself with Darwin. This is Darwin actually. I'm taking the picture with a plate, but this is Darwin himself. This is the room where the origin of the species was first discussed. So in those 10 years, a lot of things have happened. So I thought before starting with the proper talk, I would give some context on all the things that happened to me as part of the association and in my professional career. I came to the UK in late 2014, September or so, to do my PhD with Robert Scotland here at Oxford. Robert was also president of the Systematics Association. He was the president back when I joined, when I moved to the UK, and I trained as a taxonomist, as a botanist, and my PhD would be on the genus, on the taxonomy and the systematics of Ipomea and the sweet potato. We'll be talking more about that later. But yeah, this all started more or less in the last trimester of 2014. My first experience with the association, apart from the event I just mentioned, was the biennial conference held at Oxford, at Natural History Museum in Oxford, 2019. 3 days. It was a fantastic conference. It was one of my very first international conferences, and I had the opportunity to be a volunteer there. I was a volunteer. It was super busy, but it was really rewarding. And I could only remember this when at the biennial this year, a few months ago, we had more volunteers that have now joined the association since then. I think the biennial conferences that we organize every 2 years have something special. This was the advertisement, the poster for the conference. That engaged me into the Systematics Association. So for the next 3 years, more or less, from November 2015 to November 2018, I joined the council as a regular member, at the same time as I was doing my DPhil in my PhD. At the time, Mark Wilkinson from the Natural History Museum was the president, and in late 2018, that's the picture on the right-hand side, that's me giving a talk at the Young Systematists Forum. So it truly is what Eleanor was saying before, that many of us give our first talks at the Young Systematists Forum, and it's a really um, exciting moment, and this is just the beginning of many years of involvement with the association. I finished my PhD in late 2018, early 2019, and I stayed in council. But instead of being a council member, I became the newsletter communications officer. This was when we changed presidents from Mark Wilkinson to David Williams here on the left hand side. Um, and I was communications officer at the same time, more or less, as I started a postdoc in Oxford. I will be talking more about that later. What was definitory here is the pandemic. When the pandemic kicked off, everything changed in the association. We had an intense in-person presence, if you like, and since the pandemic, we've become much more virtual, much more online, with some pros and some cons. I will be going through those later. But anyway, the fact is, I was the communications officer at the time. So the time came, and I became candidate and then president of the association in late 2021. Um, Along the way, I've also had two kids, somewhere over here and here. The last one very recent, so it's been an intense time. And as well as becoming president last year, early last year, I got a fellowship position back in Spain, where I come from. So I moved to Spain, and I've been a remote president, although I've been traveling to the UK every month or every 2 months for association matters. So that's more or less the trajectory I followed in the last 10 years. You can see there's a parallel trajectory between my involvement in the association and my scientific career. Now, for the rest of the talk, I want to talk about these three things. First, I will summarize the things we've done during these 3 years with me as president. Then I will talk about the systematics and taxonomic research that I and collaborators have done during the last 10 years. And then I will share some ideas, some thoughts on the future directions and opportunities that the discipline faces and that the association faces as well. So first of all, I hope you're still there with me. I want to talk about the Systematics Association in the last 3 years. This is more or less what you have here is the number of members that we have. There was a big drop in members in 2021. I'm not sure this is real. This probably has to do with some people that had been in the member list for a long time without paying the fees. So during the last 3 years, we've done, especially Katie and Anna here, we've done an intense effort to bring everyone up to date in terms of having them as active members. And the number of members has been growing steadily in the last couple of years or so. And especially because of the pandemic, our membership has become much more international than it was before. We are a UK-based association, and we are registered as a UK charity, but now our members are all over the planet, all over the world. More or less half of our members are in countries not the UK. So that's presented us with some challenges on how to deal with the association. Researchers at different career stages, 50% of our members more or less are students. So we have researchers at very different career stages, but also from very different continents, countries, with different situations, with different needs. And also the other half of members are in the UK. So that addressing all of that is a challenge for the association, but I think we're getting there. An example of how diverse the association is becoming is on the council itself. Myself, I'm based in Spain now. We have 4 regular council members now and 1 officer that are based outside the UK. That also speaks for how global, I think, the association is becoming. I'm very proud of this. Last year, we created a students committee formed by these 4 students based in Europe and in the Americas. Because, as I said before, 50% or so of our members are students. So we need to engage with them and engage with them more, and we need to know what they want and what they think. So we thought council agreed and the AGM approved having a students committee, and the students that represent the students, the members in the council and in the events we organized, is open to new members. So if you are a student listening to this talk, or you know a student that is a training systematist and would like to join a meeting, the committee, there's a space for you. But we've also done a huge and an important effort to improve our presence and to increase our visibility. And maybe one of the main results here is that we have a new website. It took a lot of time. It took a lot of effort. I have to say thank you to Gerard Mulher, who is the website designer. Gerard spent many, many hours updating our website. Our previous website was functional, but it was old. So it needed updating. We needed something more dynamic, more up to date, basically. Peter, our treasurer, and myself were involved with Gerard, who did the bulk of the work on the website. And it's been really, really useful because it now gives us a channel or a presence, an online presence, and that's the first thing people see of us. And it's very important that we have this website there. Go and check it if you want, and if you haven't seen it, it's really, really attractive. But also because all of you will be aware, there has been a lot of turmoil in social media. So until something settles, we have to, or we try to be everywhere. Our main source of followers is Twitter, but we are still on basically all main social media platforms. And because we are all volunteers, that means that our communications officer, Anna Serra, the one in the picture before, also has to spend a good amount of time posting things and tracking everything that's going on in all the social media platforms. But it seems to be working so far. As you've seen, our members are increasing. Something that happened with the pandemic is that the Young Systematics Forum that used to take place in person at the Natural History Museum in London. We made a decision. This is the last year David was president, 2021. We made a decision to go online, fully online, for the restrictions associated with the pandemic, of course. And here you have on the top map is the provenance of the attendees that attended the Young Systematics Forum, the first virtual edition that we did. And because we had almost 300 people registered. I remember at the time, and we had to run sessions in parallel. So it was so successful that we decided to stay online. So the next years, 2022, 2023, and I don't have a map for this year, but it was very similar. We've stayed online, and we think it was a good thing to do because now we give space to speak about taxonomy and systematics to young researchers and students from basically all over the world, from Hawaii to the Polynesia to New Zealand. We have someone from Sri Lanka, I think, this year. So we have people from all over the world. It's really, really fantastic. But because we are a UK, sorry, before that, and we've done other online events. For example, this Joy of Discovery, this took place, the first edition of the Joy of Discovery took place many years ago. It's an event where speakers basically talk about a species they've discovered or taxon they've discovered and the joy of that. The title speaks for itself. We took that, resume that event again. We did it last year and we did it 2 years ago, and you can see where the speakers come from again. We have become much more global. And these are all very successful events. But at the same time, because we are a UK-based association, as I said before, we and still we can't forget half of our members are in the UK. We have to stay in touch with them. For that, it helps a lot to have in-person events. We normally have our in-person events at the Linnean Society, and we've resumed them 2 years ago in 2022 with the Founders Lecture, formerly the Huxley Lecture. We renamed it 3 years ago, and these are the three speakers that we've had. All events very successful. We've also been more involved in other activities, such as the Science Policy Lecture, which is a joint activity with the Linnean Society. The last one took place in 2022. This is the title, Biodiversity and Finance. The next one will take place, I think it's February or March 2025. So stay tuned for that. The idea here is, and the idea in this event that you see the picture, the video is on our YouTube channel, was to open a conversation between a scientist and a person from the finance world, moderated by a journalist. This event was super well attended, and it was a very nice conversation on how to connect both worlds. our work we do studying biodiversity and the finance world. It was super, super well attended. And of course, the main event in these 3 years has been the main in-person event has been the biennial, the International Biennial Conference that we used to organize every 2 years, but the last one was in Bristol in 2019. We had to stop it because of the pandemic. Then we tried to organize it in 2023 last year, but we had to postpone it for different reasons. Last summer, last June, we finally went ahead. This illustration, you see, it's the illustration that we used to announce the biennial. The conference is gorgeous. It was designed by a PhD student, Cynthia Wong, and we have posters available, by the way, if you want. So we had the in-person event. It was 3 days at the University of Reading. This was the organizing committee here. Julie Hawkins from Reading co-chaired the event. I have to say thank you to everyone because it was the activity that took most of our effort during the year, but we think it was a success. It was good that we managed to put it together and to have an in-person event again. It's because that's what keeps the I think the UK members and maybe European members together as well. So you see, we have to combine our online presence with our in-person activities, and it's working quite well. It takes effort from the officers and from the organizing committee. I haven't said thank you enough to all of them, but it was worth it. And it was a real pleasure to see that conference going. We've continued to publish some books, or at least to sign some contracts. A book has just been submitted on the new taxonomy, and we are trying, or at least discussing we've been discussing how to adapt to a changing publishing landscape. Basically, the association is not unaware of the changes happening in how people publish their work, whether people publish books or not. So all of that, it's a ongoing conversation on the association, at the association. And yes, here we are also trying to make more of our archives. The association, the Systematics Association, was founded in 1937, and we have a very interesting history. So we want to make more of those archives. We are looking for a place now to store them long term, and then we hope to be able to fund historians and archivists to work on our archives and work on the almost 90 years of history of the association. What you have here is the handwritten report of the very first meeting of the association, held on Monday, 3rd of May, at the Linnean Society. We've also sponsored events because that's one of the things we do. We provide funds to organize events. We've organized 7 events. We've sponsored 7 events, including this one. This one is the first symposium on systematics and taxonomy of tropical African fungi that has taken place. We've funded all these symposia. Some of them come with a book associated, so we will publish a book associated with those symposia. For some others, we just support the organization. And we've also continued to provide funds for research. We have this scheme in collaboration with the Linnean Society where we provide small grants up to £1,500 for research projects focused on systematics and taxonomy with a strong taxonomic component. This is the number at the bottom. You have the number of applications we've had over the years. The applications keep increasing. Sadly, the funds we have is what we can offer, but still we managed to fund almost 120 small research projects in the last 3 years. As I said before, the association is run by its council. Its council are all volunteers, so nothing of the things I've been talking about here would have been possible without them, and especially without these people here. These are the officers of the association, and David, who is the former president. All of them have been super involved and super important in all the things I've been talking about. So Thank you to all of them again. It's been a pleasure. Now I should start the next section, but because an online talk feels really lonely, can someone say, "Hey, we're here"?
I'm here.
Good. So carry on. Everything okay until then?
We're all here. We can hear you.
We're
Brilliant. Thank
here.
you.
We love it. It's great.
Brilliant. It feels lonely sometimes. Thank you. So now, yeah, I want to talk a little bit about the 10 years of systematics research I've done in during my time as a member of the association as well. What I've been talking, I'm going to be talking, I'm giving the talk, but obviously this is the work of many people. Most of the things I'll be talking about is the people that have been in Robert Scotland's group at one time or the other. Most of us continue to work together in pending activities, and most of the things I'll be talking have been possible. I will be talking about have been possible thanks to these chaps. But now I've also have other projects going on involving all these people at the bottom. They will pop up again during the talk. And I will be focusing on two projects, on the two main plant genera. I'm a botanist, so the two main plant genera I've been working on. One is the genus Ipomea, and the other one is the genus Acalifa, a convolvulaceae and an Euphorbiaceae, both of them quite big. Ipomea, a mega diverse genus, some 810 species or more probably. Acalifa, over 400, 430, 450. We are working on that. I will be talking about both projects. Um, and what is in common between the two projects is the idea that at the center of each of those projects is a taxonomic monograph. Basically, a systematic study of a taxon at a global scale and integrating morphological, ecological, and molecular data. And we think, although when people hear about or think about the taxonomic monograph, may think of something that is old, outdated, or that used to be done in the 19th century or the early 20th century, we think taxonomic monographs have a real impact on the science we do, and not just on the taxonomy, but on many other disciplines, as you will see in a few minutes. So it all started with this idea of a foundation monograph of Ipomea, Robert Scotland, and John Wood, the second person uh, in this picture. They came up with the idea of a foundation monograph as a way to accelerate the pace of taxonomy. I suggest you go and read this very short piece if you want to learn more about the idea of a foundation monograph, what a foundation monograph is. The idea is that monographs take a very long time to produce, normally, or used to take, often the lifetime of a researcher. And in the current scientific landscape, scientific context, that's not feasible anymore. We have to accelerate taxonomy, and if we want to go for monographs, we have to accelerate them. How we do that? Working at different levels and with multiple people working in parallel. Looking at we mostly work. I will delve more into this. We work mostly on herbarium specimens, and we do all the morphological studies, but we also do um, molecular stuff, and we ask and try to answer questions beyond the taxonomy for which the taxonomy is necessary. I recommend you read this very short piece. So I came to Oxford in 2014 to work in Acalifa, and the overall project of the group, of Robert's group, was to produce a foundation monograph of Ipomea in the Americas, 425 species. It took us 6 years of work, I think it was. It's here you have it. It's an 800 pages book, and it was submitted in 2019. It was published in 2020, the very same day that the lockdown started in the UK. So that was unfortunate, but luckily the monograph got traction. It was the four of us working in parallel, mainly Beth and I doing the molecular stuff, and John and Robert doing the taxonomical stuff, and then everyone together working on species delimitations, discussions, etc. Herbarium work, field work, everything, and other people involved as well. Um, this monograph obviously produced an important improvement on the taxonomy, on the knowledge of the genus. We described, as part of the monographic process, we described 75 new species, identified, classified, and organized all the synonyms, over 3,000 names published for Ipomea, around 69% of them synonyms. That's in line with other big genera, especially tropical plants. And we produced many tools for people to use them. We produced identification keys. We produced illustrations, photographs. We've generated distribution maps, phylogenies, molecular phylogenies using DNA barcodes and somewhere sequencing, but also using genomic scale data. I won't be talking much about those now, but we've done those and we've answered all the questions. We have a database with more than, now it's over 35,000 curated specimens, so specimens that we ourselves have identified and are a resource for people to check if they want to check. We've produced sequence data for many specimens, and along the way, as part of the as well as the monograph, we've published other region-specific or taxon-specific articles. But this is where I want to talk about how producing a taxonomic monograph, the results exceed the taxonomy. And this is where our monograph became very powerful. Thanks to having a good taxonomic knowledge of the Ipomea, thanks to producing a monograph or working on a monograph, we were able to address some questions that had long been debated with unclear answers. And one of them was the origin of the sweet potato. The sweet potato is Ipomea batatas, that's the storage roots. It's a member of the sweet potato of the Ipomea genus. It's a member of the Ipomea. And it had been the matter of discussion. The origin of the sweet potato had been the matter of discussion for 200 years. But looking at the literature, most studies had, in essence, all studies had conflicting results. And all species in the group of sweet potato wild relatives, crop wild relatives, some 16 species, all of them have been proposed, have been suggested as progenitor of the crop species at some point by some author. Obviously, a crop can hardly have 16 different progenitors. So what happened is that all existing literature was flowed with taxonomic error, basically. The specimens in this group are difficult to identify. They have overlapping morphologies. They have overlapping distribution areas, overlapping ecological requirements. So there is a lot of misidentified specimens. I don't have the diagram here, but throughout the monograph, around 50% of the specimens that we studied had a different name at the end of the monograph. So almost 50% of the specimens that we studied were re-identified. This obviously affected our studies on sweet potatoes, where because for the first time we had a very clear idea of what were the relatives of the sweet potato, what were the species boundaries, and what could and what could not be the putative progenitors. But because normally what people would do is do the taxonomy, sort this out, and move on to another topic. But in this case, beyond taxonomy, we carried on working on the sweet potato because resolving the phylogeny and the taxonomy of the species closely related opened new questions. Now, sweet potato is a hexaploid, and we had identified the species that is sweet potato's closest diploid relative. But the tetraploid, the closest tetraploid relative, was still missing. And we're still not sure whether sweet potato is an autopolyploid or an allopolyploid. That's something I'm working on at the moment. But at the time we finished this taxonomic study, we had identified the diploid, but not the tetraploid. We had some hints. So we applied for a project. That was my postdoc, where we had identified some populations of a tetraploid plant in coastal Ecuador and Colombia. traditionally identified as feral, as wild sweet potato. But when we look at those, those samples were morphologically different. In the phylogenies formed a monophyletic group sister to a monophyletic sweet potato, and had all the characteristics of being of wild provenance. So thanks to all the work we had been doing before, we managed to identify and discover this entity that had been there in front of everyone for quite some time, and propose that it's sweet potato's closest tetraploid relative and the most likely direct descendant of its tetraploid progenitor. But we also didn't stop there, and we thought if this plant is there, it's important to study it. So we continued. Remember, the monograph was published in 2020. This article describing the species was published in 2022. And only last year, 2023, 2022, sorry, we did fieldwork in Ecuador to characterize the species. And what we found was that the species wasn't restricted to an area in northern Ecuador where we had seen herbarium collections. The species was actually widespread in Ecuador. And some of those places have been visited by previous taxonomists that were also experts on Ipomea. But because our taxonomic knowledge and our global scope, the global scope that the monograph gives, we were able to identify them that are morphologically and molecularly different from sweet potato. So here you see how the results of a monograph exceed the taxonomy itself and get into crop wild relatives, food security, crop origin and domestication, etc. So this is now more or less the landscape we have. We have a hexaploid sweet potato, Ipomea batatas, and we've identified with very good certainty that Ipomea trifida, a circum-Caribbean species, is its diploid progenitor, its diploid closest relative, and Equatoriensis is its tetraploid closest relative. But we've also explored all the questions during this time, and also thank you to the monograph. We, for example, discovered and dated and studied 2 rapid radiations in Ipomea centered in the Chaco region and in Central America and in North and South America. 2 rapid radiations that are equivalent to some of the most iconic radiations in biology, and this was thanks to having a good knowledge of the taxonomy of the group. And we've also done, I won't go into much detail here, but for a long time, sweet potato was an iconic species for its storage roots. But the fact is that many other species in the genus Ipomea, this is a phylogeny, develop storage roots or underground organs as big as those of the sweet potato, some of them even bigger, and also edible. And there are records of where those species have been used in the past, even though they haven't been cultivated. So we've also been studying them. We are still studying those across their distribution, trying to understand what promotes the origin and development of storage organs in the genus. Obviously, a monograph is not the end. The monograph opens the door to much more research. One of the things it enables is the discovery and the description of new species. And since we published the monograph, there have been at least, the monograph was for the Ipomea species in the American continent. There have been 7 species described, some by us and some by other people. I will go back to those. So the monograph also enables the identification of still hidden diversity. Even though we think we have a good knowledge of the diversity of the genus in the continent, there are still some species pending to be described out there. And also because of the monograph, we've had a lot of discussions and have devoted a lot of time to discussing some of important theoretical concepts, from how to classify big genera when there are no diagnostic characters, how to identify cryptic nodes, how long it takes to discover a species, or what is a species? How do we taxonomists that work with a species, how do we identify a species and delimit them when the amount of data we have is limited and the data itself is fragmentary? And we've discussed this in this and other papers. And just last week, this article came out in Q Bulletin where we actually explain the methodology of how we did a monograph of hypomia, a methodology that hopefully others will use in their groups of study. Some, for example, how to make a decision on what species recognize or not, how to start, where to start. That's always very difficult, and that always makes people refrain from starting a monograph. We try to explain everything there and also discuss some of the other aspects I'm discussing here, some of the consequences and some of the other activities that result from the monograph. But again, we've been involved in other studies, and I think this is important, that go beyond taxonomy. We've been involved in, for example, these studies looking at how climate and climate change can affect or can alter the distribution of certain species. We have a consultative role in different countries for different works, where we have active collaborations going on as taxonomists, but also beyond. And there have also been some spin-offs, and this is one of them, of that research. As I mentioned before, there are many species in Ipomea that develop underground storage organs. This is a project I got with Javier Fuertes here in Madrid when I moved 2 years ago, where we want to study certain species in their habitat and understand what are the similarities and differences in their storage organs. We are doing field work, we are doing herbarium work, we are doing molecular work, and we are growing plants in the greenhouse, doing anatomy, doing all sorts of things that go beyond taxonomy, but that taxonomy enables, because now we have a good knowledge of the species and a good delimitation of the species boundaries. And this project started, by the way, with this species, Hypomia noemana. That's one of the species that we described after the monograph. And this species has edible storage roots. It's probably 310, so we are also studying that species in detail. And that project is the two of us, Ivalu from the Instituto de Biología in Mexico, and Enoch from the San Marcos University, and other people working in our groups as well. Now, that's Ipomia. The work we did on Ipomia got us thinking more and more, as I've been doing throughout this talk, on what's our role as taxonomists on science. How can we contribute? How can we make taxonomy highlight? How can we highlight taxonomy beyond taxonomy itself? Sorry. So the other example I want to bring here is a project I lead here in Spain. We started last year, a year ago, and this project the idea is to produce, same as with Hippomia, a monograph of a Acalypha, in this case, in continental Africa. Acalypha is a genus, although I did my PhD in Hippomia, I've been working on Acalypha for a long time, since my undergrad with Jose Maria. We were mostly focused on the Americas as well, but now we are giving it a global approach. And the idea is to incorporate all the things we've learned thanks to the Ipomia project and all the things that occurred after it, and integrate them in this project. This is an overview of the project. I won't go into much detail, but the idea is that having taxonomy and systematics at its core, where we combine morphological studies and DNA studies using herbarium specimens, also fieldwork, as the main source of material, we do much more than that. We do things that are normally done as part of a taxonomic study, but that we think can be done and should be done alongside the taxonomy and that only the taxonomy enables. We do micromorphological studies, we are doing conservation assessments, we are studying, I will mention it in the next slide, the medicinal properties of some of these species, and we are planning to do also species distribution modeling, chemical studies, etc. The idea here is that taxonomists, most of us here are taxonomists, that we can also lead projects that ask questions beyond taxonomy. So, as I said before, or as I was planning to say, we chose Acalypha first because it's a genus we were familiar with, but also because it's a big genus, and it's at least 60 species in the group are known to produce compounds of ethnobotanical or medicinal interest. And some of the species are being studied through experimental essays to study these compounds. What we found is that literature on ethnobotanical and experimental research on Ipomia accumulates as much useful information as useful error. This diagram here, very briefly, is all the uses recorded. This has been done by Lucia Villascus, a PhD student with me, where we categorize and classify all the uses of the plants. And then what Lucia has been doing is she's been looking at the taxonomy and how the taxonomy is presented in those studies. And this is quite so. Has it been cited properly? Can we identify the plants? Truly identify the plants or the species based on the information on the ethnobotanical or the experimental papers? This is important because experimental research moves a lot of money. So there is money being invested on this. Can we rely on the results? What Lucia found, this is study in progress. We hope to have it ready very soon. Is that around 25% of the articles provide incomplete information that doesn't allow to certainly confirm that the species they mention is the species they are truly studying. So basically, 25% of the species of the articles published on experimental studies and ethnobotanical studies in Acalypha don't allow to confirm the identity of the plants because they don't give the taxonomic information correctly, or they don't provide voucher materials, or they simply say Acalypha sp. So this is a waste of money. We think by having a proper taxonomic framework and a proper taxonomic knowledge of the group, we can and we should improve how that money is spent. And also having a good taxonomic knowledge will allow to confirm if the species that people say have certain medicinal or pharmacological properties truly have them. We've been doing more things. We've been doing micromorphology, as I said before. We are doing a lot of work on herbarium materials. And because we are taxonomists and our primary focus is taxonomy, we are working on it. So a nomenclatural review of the group is already in review. So we hope to have that ready soon. All of this within a year since we started the project. And because we think publications nowadays are changing, a key component of our project, of our Acalypha project, is the project website, where we can publish information much faster than we do in traditional peer-reviewed papers. All the information on the website goes to papers, but here people have access to all the nomenclatural information up to date and available, access to over 40,000, I can't remember how many specimens, many of them identified by ourselves, and now we are incorporating more information on conservation uses, the phylogeny of the phylogenies that we are producing, everything that to help users access the information as soon as possible and in parallel to the traditional publications. Now, can I confirm you're still there?
Yes.
Still here.
Thank you. So that's an overview of what I've been doing, or some of the things I've been doing over the last 10 years. I hope it all made sense, more or less. The last part, the last few minutes, I want to talk about the future directions and opportunities that I see the discipline could take. This is just some ideas. There is no silver bullet, but this will be my ideas on it. Also on the future of the Systematics Association as a body, as a professional society representing systematists and taxonomy, and it has some relationship with a talk I gave at the biennial last June. It could be future directions and opportunities, or it could be bringing taxonomy back into the spotlight. So we now have access to more data than never before. Biodiversity data, occurrences such as these ones in GBIF, data in GenBank, iNaturalist. We have access to more data than never before. And this gives, maybe not people in this room, in this lecture today, but this gives many colleagues in other disciplines the idea that we have a good knowledge of the planet's biodiversity. But still, there are thousands of species yet to be described, especially in tropical plants and in insects and other groups. We've seen that herbaria worldwide that are the main source of information for those global databases accumulate a lot of error, up to 50% in some tropical plant families. And we know that despite having so much more data accessible now, it still takes decades to accumulate a basic knowledge about the species that enables any subsequent study. And here, when I say basic knowledge, this relates to a study we published in 2020, is we counted how long it takes to accumulate as little as 15 correctly identified specimens for the species in the 20 largest families of flowering plants. And it takes decades to accumulate as little as 15 correctly identified specimens. In a context of climate change, in a context of biodiversity crisis, it seems like a lot to have a proper basic understanding of the species of the world. Now, I was looking at how often people use the word taxonomy and systematics, and it's interesting. This is a Google Books Ngram tells you how many times a word appears in the books scanned by Google. It's paradoxical that the time when people started talking more about biodiversity coincides with when started talking less about taxonomy and systematics. And I think it has to do partly with how the biodiversity studies are moving towards more ecology-based studies, if you like, or more climate change stuff, or other more applied science, if you like, although I don't like that word. It coincides with, it gives that false impression that we don't need taxonomy. But as many or everyone in this room knows, taxonomy for many groups is not robust. We don't have a robust knowledge for many groups. There is much more work still to be done. And we've built already, we are building already a massive infrastructure of biodiversity data and biodiversity results that rely on a very fragile taxonomy. So the greatest obstacle to discovery is not ignorance, it is the illusion of knowledge. And I think that's what's happening with taxonomy in some cases, or in many cases. Obviously not for people here. So taxonomy faces important challenges. Some of them are old, and people much older than me that have been here for much longer have been warning about this before, and many times, and much better than me. Some other challenges are new. As I mentioned before, the misconceptions from colleagues in other disciplines, that's damaging, I think, the the systematics and taxonomy as a discipline, the lack of support from stakeholders and managers and scientific societies that divert from taxonomy to pursue more trendy topics, topics where the money is now. That's happening. There is the detachment, ongoing and worrying detachment between the scientists and the collections, with several different institutions in around the world either getting rid of their collections or separating them from the scientists that use them the more. Obviously, the limited funding, I have nothing to tell you about that. There is the impact factor, publishing trends. We have problems competing there. Traditional taxonomy has problems competing there. And then there is the challenge that is also an opportunity. There is the big data, the machine learning, artificial intelligence, and genomics, and all the new technologies that are being developed. A challenge is to incorporate those, because they won't disappear. We have to incorporate those in our work. That's where we argue that, contrary to the impression that they may give, my argument is that monographs are the future for taxonomy, because we now have the resources and the people to produce them in a short time frame. We have to work at an evolutionary framework. Obviously, we have to work at a global scale and as a team effort. Monographs can no longer be a one-person activity. We have to work as a group. But if we do that and we embrace technology and we use all the resources we have and we keep accessing herbaria, obviously field work as well, and combine all these things, as I've showed before for Ipamia, and I hope to show for a Khalifa, the impact that a monograph can have goes much, much beyond, far beyond taxonomy. Now I think we have to play the game. I don't really like that sentence, but it's what we have to do. We have to defend taxonomy actively, not just talk about how damaged we are, but work towards make the situation change. The rest of the scientific community is not going to change it. It has to be taxonomists who change how people perceive taxonomy. We need to increase visibility, the relevance, the integration with other disciplines. It will be much easier if we are the people that lead the projects. For that, we have to be ambitious and ask global questions. I think, what questions do we want to answer by resolving the taxonomy? Resolving the taxonomy, you don't have to tell me, is obviously a goal on itself, but in the current research funding landscape, we have to ask global questions, and if we don't ask them, other people will. So if it's led by taxonomists, if those projects are led by taxonomists, we will make sure that those questions are answered from a robust taxonomical framework. And then we also have to go and talk to other audiences, because we talk a lot to other taxonomists, but I think it's time, as we've did with the science policy lecture and other talks we've done, we have to talk to people in other disciplines, in other scientific disciplines, and also in other worlds, people that managers, stakeholders, everyone that can make a decision on where to put the money. And we have to present our research in other fora. Before my final slide, I want to bring up this quote from an interview to Robert Scotland a couple of years ago. We have to trust that what we do is fundamental and is good science, and we have to advocate for it in all the different places where we have access to. Now, this obviously links tightly with the systematics association, with us as the association. And I want to finish here, because same as the discipline, the association also faces certain challenges and also some opportunities. And I've been mentioning some of those before. I'm going to just list them here again. We are much more international now, but we have to embrace that international membership. It's really diverse, people at different stages in their careers, and an enormous potential for the association, but we have to embrace them. We also have to strengthen links with the UK membership, still obviously the majority of our membership. The pandemic damaged us a little bit, but we are recovering little by little, and having more in-person events, it's truly the way to go. We need to retain members. We need people to realize that the Systematics Association is the place to stay and to help and be the voice. We need to increase our visibility. We are working towards that. We need to actively involve the newer generations because they will be here for a long time and they are already exposed to all the new technologies, to all the challenges, to everything. And half of our members, as I said before, are students, so we need to actively involve them in the activities we organize. We need to adapt to a changing publishing landscape, as happens with taxonomists. The association is no different. We publish our own volume series, but we need to rethink on how we do what is our publication strategy and whether how to reach beyond all the publics. We have to foster relationships with other European associations. We've been doing it a little in the past. We have to do it with more commitment. There are other, or even outside Europe, there are other systematics and taxonomic associations out there. And the more we are, the stronger we will become. And again, we have to increase our impact beyond systematics. I think it's the only way for taxonomy and systematics to come back to the highlight, to the spotlight. For that, the association needs members. This is my second to last slide. The association needs members, and the more we are, the more we can do. And the association relies on its members for all the activities we do. We are all volunteers. So if you are not a member yet, please consider joining, and your students and your colleagues, talk to them. And just to finish, I wanted to finish with a smile here. These are because I think this talks about how successful we can be as an association. These are some of the comments we had as feedback from the Young Systematics Forum we organized a couple of weeks ago. So that's it from me. Thank you all very much.